Thank you, John. And now we will invite Ana Estival, Manuel Cobo, and Ana Laura Ortega for the panel discussion. So perhaps we can start.
I'm sorry. Yeah, I have a question for Dr. Garcia and Dr. Rek. So in the setting of EGFR mutant patients, as we are moving combo to first line, where is the perfect place for ADCs after FLAURA2 or Mariposa? What do you think?
This is a good question because in the first line, we are thinking that now a combination maybe is becoming the standard of care. We don't know exactly which is the combination, and now we are seeing also the trial in this setting. I think that this will be based also on the toxicity profile, because these patients, they live for a long time, and sometimes what we see of the ADC combined with osimertinib in the first line, maybe we will see in the end if it is the balance between toxicity and activity will be in favor of the combination.
I would agree. I think this concept to have an ADC targeting ETF receptor is somewhat compelling, but we need to see the clinical activity in larger populations, and I think we are not now at the stage to compare this to OSI-mono, so we really need to compare this to other combinations, whether there is some additional benefit. And the big problem is really how long should the patient receive the ADCs, because this is something we do not know. And there are some chronic toxicities associated with these ADCs which we have to monitor, and I think we may not forget that these are patients who are living for years, and we have to keep on the tolerability, the quality of life, and the maintenance of this life value. So I think multiple questions. In principle, I would say, yes, it could be a new combination, but it has to be confirmed.
You were talking about combinations of the ADCs drug. What about any potential role of the anti-angiogenic combination with this kind of drugs in order to get a higher payload delivery? Is there any role of that?
Well, theoretically, yes, but so far I do not know any data, but I think it's a very interesting concept. In particular, we will hear a lot about these bispecific antibodies which are targeting anti-PD-1, anti-PD-1, and anti-VEGF. So this could be an interesting concept. There are plannings in place, I know this, but this is something which needs to be confirmed by clinical data as well.
And any additional worry about potential toxicity, because maybe add, as we talked yesterday, it could be an issue.
Yeah, the interesting part of these anti-PD-1, anti-VEGF antibodies, when you look on the typical anti-angiogenic side effects, the incidence is quite low. So when you look, for example, on the HARMONY trial in squamous cell cancer, and when you look on the eligibility criteria, there were patients with centrally located tumor, even history of hemoptysis, and other clear exclusion criteria, where I never would have dared to give an anti-VEGF compound to a patient with squamous cell carcinoma. And when you then look on the rate of adverse events, this was surprisingly low. So the big question is, what is really the anti-angiogenic activity of these compounds? And this is something we need to know.
One question, respect the importance of the receptors in the ADCs, because we have learned, for example, with HER2 mutation, that trastuzumab is important not only to introduce the payload, is to control the receptor. To control the receptor, to internalize the receptor. The receptor is important. There is important when it's mutated for all the phenomenon of progression, survival, etc. And perhaps the good response of the trastuzumab when there is HER2 mutations is not only for the effect of the payload, is the effect to destroy the receptor. So what's your opinion that perhaps focus the development of ADCs to receptors that are important for the survival of the malignant cells. For example, TROP2 is very expressed, but perhaps is not a receptor that is important for all the effects of survival and progressive disease. And perhaps focus to another class of receptors like the integrin B6 or MOOC or another class of these class of receptors that are important to survive.
I completely agree, but I think I would give this to Marina.
No, I completely agree with you. I don't have anything to add, but I agree. Sorry.
Yeah, I think it's now because the story is now getting a little bit more complicated because we have more and more ADCs not only targeting one single target. So I think the future is more in having this bispecific or even tri-specific ADCs which are targeting several key structures, and then it will become very difficult to sort out which is the relevant target, which is the relevant antigen for the tumor survival and the tumor proliferation.
And,
although, if I can add a comment
there is
here.
also the activity of the payload that I guess has a lot of importance. There are all the transporters that maybe are underestimated that can import the drugs into the cells. So I don't think it's just a matter of payload and the monoclonal antibody.
Yeah, I have actually, if I can have a comment here. So initially the field was very much focused on the actual protein that was targeting the antibody. Now my feeling is that the payload is gaining more and more importance. For example, we have ETHA-Brand data, this specific EGFR HER3, showing quite remarkable activity in small cell lung cancer, which we wouldn't predict EGFR and HER3 being that important in that situation. So maybe the payload is more important than the target, maybe.
But when we take on the possible mechanisms of resistance to the ADCs, our enrichment, because not only because of the post-transcriptional inhibition of the receptor, there is two problems with the lysosomes, for example, and the mechanisms of resistance receiving the payloads are too important. It's certain that we need to develop payloads with every time more and more activity, but sometimes the mechanisms of resistance of the payloads are too important in my
So in line with this, my question for you two would be, are we actually learning anything from every single failure or negative clinical trial in this, in the way we are approaching the strategy to develop ADCs in non-genomic-driven non-small cell lung cancer? Are we using biomarkers, mechanisms of resistance?
So the problem is, I think we have to learn. Currently, we are a little bit behind because when you look on the classic phase 3 trial, this is in a maximal unselected population because there is the interest to have a big population benefiting from a new approach. And when you then look on the biomarker approach, it's a retrospective, it's an exploratory approach like the work that has been done by Marina. And this is always something which has to be confirmed prospectively. And this is also a matter of time when we are there to have a really predictive biomarker. And I think this is something we should consider in principle when we think about new concepts of treatment, that we need some more intensive biomarker identification before we go to this prospective validation. And I think this was a little bit disappointing when we started with the ADCs because we said, well, now we have a targeted chemotherapy, we just have to look for the expression and then we are there. We learned from breast, this is not the case. We learned from lung, it's the same. It's more the presence of the antigen probably, but I think now we have to reconsider more in principle our construction of prospective trial and the integration of the biomarker research.
Oh, and I think also if you look at the data of the QCS and MR, that is the biomarker, they can divide the population that is responding and not responding, but still for about 50% of the patients, also if they are QCS and MR positive, they don't respond. So this is the part that I think we should try to understand.
So, one question, as we move to cell therapies, right, we're developing new cell therapies, CAR T, TILs. The challenge that we face, and I have faced, despite my patients being very young, is that they get to a point they're too sick, and then they're not able to get into the clinical trial. And these myeloablative regimens that you need, which brings flashbacks to all of us doing bone marrow rotations as a fellow, when is the precise or adequate moment to start thinking about enrolling these patients and cell therapies, clinical trials? Because I believe, like in my institution, we're trying, but it's hard. I have 2 patients that they enrolled, but they couldn't wait for the cells. One passed away before. So that's what I struggle. When is the right time to prepare to get them there?
Yeah, this is the critical challenge that we are facing in clinical development of these therapies for solid tumors. And this limitation is actually also reflected in the Moffitt Cancer Center trial, where half of the patients actually couldn't receive the therapy because they were progressing during the T-cell product preparation, for example. So, I mean, in theoretical terms, probably the best opportunities perhaps in the frontline setting. But the problem is that in the frontline setting, you really need to be sure that the toxicities are balanced with the efficacy. And for example, in melanoma, when you already have a trial in refractory patients, there are now clinical trials in the frontline setting. But people, if you talk to the physicians treating melanoma, they also have doubts because pembro monotherapy can produce very long responses, and you are adding a T-cell product here that there is some issues about toxicity. So it's a really, really challenging situation, and I don't think we will have the answer. So the sooner the better to start preparing all the cellular product, but this has some challenges in terms of clinical implementation.
Yeah, I think this time I agree with you because it's because I think when I look on the sites in my surrounding where we have cellular therapies, they are always at the very, very end of any treatment line, and this is not appropriate. So if we do really think this is a concept of interest, and even when you look on the data of pancreatin, there actually have been some patients who had just received nivolumab as a first-line treatment. And I think we really should strengthen our efforts to keep these trials in earlier stages, at least in talking to patients, that there is the opportunity and to get them on these protocols as early as possible. I think it will never work if you have this treatment on the far end of the therapy line.
So there are now this technology evolving, particularly like in vivo CAR T cells, that is maybe solving part of this production issue and the bridging between, right? So do you think that is going to be also a potential strategy to use TILs, and not only TILs, but actually engineered TILs? I mean, you show some data of TILs able to actually produce T-cell engagers and immune checkpoint modulators and so on.
Yeah, I mean, definitely this probably would solve this problem of production, and you will get, let's say, fitter patients into receiving these therapies, and I think this is an advanced. My concern there is that how do you really control for the doses of the product that you are giving to the patient? I'm not sure about whether we are able to control that. And probably with more advances in technology, we will be able to do that in the future, and hopefully this will be a way to solve some of these limitations. But I have concerns on how do you control the actual dose that you deliver to the patient.
Actually, today, honestly, we don't know very well the dose of this biological we are giving.
Okay. The last question, please.
Thank you very much for the presentation. I mean, with the ADC, you showed that there is so many options now to arrange the antibody targeting the target and the payload. And it seems like it requires time to get data, mature data, and sometimes when we get the data, the control arm is not updated anymore, up to date anymore. So the question is, like, do you think clinicians, AI, machine learnings, and pharmaceutical companies should sit very early on to maybe choose the best candidates and focus on these candidates to get data very quickly, or is there a benefit to actually let all the clinical data for the potential new ADCs to rise and get some novelties?
So I think it's a principal question, and it's a very good question, because when I was starting in thoracic oncology, we had the phase 1, the phase 2 as the signal-seeking trial, and then we had the confirmation with the phase 3. That took a couple of years. Now we just have the phase 1 with the dose expansion, and we are moving to the phase 3. And we have the problem that we cannot validate a number of very promising phase 1 results in phase 3 trials. So personally, I think we should stay a little bit conservative with just validating the clinical signal with valid numbers of patients. But it's a real challenge. It's a task, because the speed of drug development is moving on, and it's moving on. And I do not have a good answer to this, but it's a very important question.
Thank you. Unfortunately, we need to close this session. Thank you for the presentations and the discussion.
There is no break.
There is no break.